Amphibia and reptilian type.
Okay, so we're continuing on our subphylum Vertebrata. Before the holiday, we did what I'm calling the Agnatha, even though that's not really a technical name. You should know what that means for the test. And when I have a lamprey or a hagfish, you should know that that Agnatha basically means jawless, or there are no jaws. Anyway, the rest of these groups are what are generally called the Gnathostomata, and I think I convinced you that the jaws evolved from an exaptation of the first two gill arches. In the shark, you can see it really pretty clearly, and then in the fish, bony fish, it was a little bit more difficult to see where that jaw was technically oriented. The TMJ, the temporomandibular joint, the cartilage that connects your jaw to your skull, basically is the mandibular cartilage that made up the shark's front jaw, and that continues on with the rest of these organisms. So we're going to cover today is the amphibia, the reptilia, and the aves. So we have kind of the whole gamut here of these, and we're especially going to look at skull structures in the reptilia and show you some interesting features there. But let me just go ahead and do a review again of what the fish condition was. And I think what we did here was something like this. If we look at what the fish had going on, or I guess, let's go ahead and do a review of the shark. Remember, the shark was basically, here is the shark cranium, here was the mandibular arch and the hyoid arch, and then we had 5 gill arches. This was basically how the shark's cranium and jaws and gills were kind of oriented. So we had the mandible or mandibular arch, which was number 1. We had the hyoid arch as number 2. I'm going quick because we already did this one of these. And then these are gills, number 3 through 7. And remember, way back to the lamprey, basically there were just 7 gills, and that was kind of how they were put together. So with the bony fish, I think I did something like this. We had the bony fish look something like this. So there's our kind of our skull of our bony fish. I think I said that we have a cranium here, and then we had our number 1 is our TMJ, our temporal mandibular joint, was the joint between the maxillary bone and the mandibular bone. And then we had a hyoid bone, which is called the ossicle. Number 2 was the ossicle, which is the hyoid. Number 3 was the operculum. And then there was 1, 2, 3, 4. There was number 4 through 7 as the gills. So that was kind of how the fish was put together. Was the same arches are present, just they're reduced and maybe more focused because we have a replacement of everything by bones as a skull. So this is our cranium. And this is the cranium here. Okay, so that's what we already covered so far. Let's look at the amphibian condition here, or the amphibian jaw. So we saw this this before. Here's the amphibian. So what we have going on basically is really it's just kind of like in my opinion, it's like a modified fish. It has kind of the same appendages and conditions as the fish. We have a skull, which is a combination of cranium, jaws, and gills all put together. We have pectoral fins and we have pelvic fins and attached to a vertebrae or a backbone. But in this case, you can see that in a larval or a salamander that's retaining juvenile features, it kind of has very much a fish skull. And in fact, it still has gill arches that are present there. We have pectoral structures that are basically for crawling or walking on land, but essentially they're made up of the same sort of bone structures as the at least lobe fin fishes are. And then the pelvic girdle. So, essentially it's just kind of like a modification of structures that were already there. So let's take a look at an amphibian skull here. I do have a salamander's skull here on this piece of paper you want to take a look at, especially on the bottom side, as you can see the gill arches that are present. So let's go ahead and see what we got going on. This is like a salamander skull. Just get this going here. Okay. So we have the same basic idea. We have a cranial bone here. Cranium on top. It's actually a bone. The whole thing basically is a cranium in terms of the skull, but that front plate is basically the same as it is in the fish here. The TMJ is still present, and that's still present even in you. The TMJ, the temporal mandibular joint, is the cartilage that cushions the maxillary bone and the mandibular bone there. Okay. The ossicle, though, in this case, you can't really see it in our salamander skull, but we will see it in our frog dissection today. The ossicles are now converted into the inner ear bone. And this is number 2, which is, they're called the ossicles in ossicle. In vertebrate biology, but actually this is what you would call the stapes, because it kind of resembles a little stirrup, like a horseback riding saddle stirrup that's attached to the inner ear bone. So this stapes here is associated with the tympanum or the eardrum on the frog, and so essentially the highway bone, the second jaw, becomes the ear bone, which is why your hearing is so closely associated with your jaw. And in fact, if you have ears that are plugged up because you've got a high elevation or you fly in an airplane, one way to clear your ears is to kind of open and close your jaw. And what that does is it opens up your eustachian tubes that allow for your equilibrium. But your jaw, your TMJ, and your ear bones are basically so close together. And the reason for that is because they're originated from those first 2 gill arches, basically from sharp condition. Then the operculum goes away, so number 3 is gone, and number 7 is gone. And what we have in the frog here is the salamander here in a polywog frog in the case. But we have number 4 through 6 as gills. So number 3 and number 7 are vestigial; they go away. And so check out this little salamander skull here. You can see there's 3 gill arches basically coming off the backside of the salamander skull. So that's really kind of neat to see that comparison. But that, eventually, basically it's just a modification of the fish condition. Okay. All right, so for amphibians, we have basically 2 different basic groups of amphibians. What we consider to be toads and frogs are basically the groups called Anura. And they're called the Anura, I forgot to bring out my frog skeleton here. They're called the Anura because the term Anura means no tails. I'll write this down here. Here's our little frog skeleton. So let's see here. So Anura means is basically the frogs and toads. And what Anura means, this would be good to know for the test, means no tail. And that's because frogs and toads, as an adult form, the tail, there is a tail present, a tailbone, but it's actually pulled inside the pelvis. And so the tailbone is internal. And you can see it here on the frog skeleton here. The tailbone is there, but it's basically inside of the pelvic girdle for support. And then the other group here would be called the Caudata. These are order names, or orders below class of the vertebrates. And this would include the salamanders and newts. So, and what Caudata means is tail. So really, the true classification is we have tailed amphibians, which are the salamanders and the newts, and we have the no-tailed amphibians, which are the frogs and the toads. But the name frog and toad and salamander and newt really don't mean anything. It's a polyphyletic group that I guess if it's more aquatic, like if the adult stays in the water, it's called a newt, and if the adult moves more on the land, it's called a salamander. So the more aquatic adults are called newts, and the more terrestrial adults are called salamanders. But there's actually no relationship kind of among the groups that are called newts and salamanders. These are just common names that are used for their habitat. Same with frogs. Frogs would be a more aquatic adult, associated with ponds or rivers or tree holes. And then toads would be more of a terrestrial adult that can tolerate more dry habitats. All of these, though, have to go back to the water for reproduction. The larval stage of a salamander, here is an example of salamander larvae with the external gills, pretty much resembles a larval stage of a frog or toad. And so they need basically to have that aquatic stage in their juvenile stage to survive. Okay? So what I have here, what would be on the test would probably be, I have a couple salamanders in jars. They actually have letters on top indicating they've been used for the test in the past. Okay, I use either one of these. And then we have a toad here in letter A there. So essentially, I think what I'd probably ask you is, the class is amphibia, basically. So amphibia, they're amphibians. And the group basically would be Caudata versus Anura. That's the technical names. These names, frogs, toads, salamanders, newts, are just common names that don't really make any sense. But they're referring to basically the ones with tails or without tails. So I think that would be a cool thing to know, the basic grouping. And then I think, I don't think I'm going to have any questions on the skull for you, because it's really kind of hard to label that little skull. So probably would be on the actual frog section that we're going to do, is what I'd ask you. Let's go ahead and we'll cover the frog dissection at the end, but I want to jump to our other groups to talk about their skull features, because they're kind of interesting as well. So we'll come back to the frog dissection that we're going to do at the end here. Let me just go ahead and jump to what it means to be a reptile versus a bird. And this is a really interesting, confusing thing. So this whole group of what makes up the reptiles and birds and the mammals, let me go back to our classification list here. So this whole group, Reptilia aves, which are the birds, and the Mammalia, they're all classified based on having an egg that can survive on land without water. So on Thursday, I'll talk a little bit more about this, but there is an amniotic egg, or an amniote egg, that enables these to no longer need water for the larval development. Instead, the larvae, or the immature, develops inside of a shell, an egg, that has that water environment. So you can think about, pollywogs and tadpoles swim around in the water to develop and mature. These organisms basically have their embryos swimming around in an egg to mature. And it's that amniotic egg. We'll cover that on Thursday, the details of that. But that's what links all three of these together, the reptilia, the aves, and the mammalia. So anyway, this is a confusing thing because what's interesting is that the reptilia and the aves especially are like a mixed group. And I'll show you what I mean here. Is the reptiles, what we consider to be reptiles, everyone kind of knows a lizard when you see one. So here we have some lizards here, and we have a snake here. And that's pretty clearly what you would call reptiles. But reptiles also include some other things. Namely, we have turtles. So turtles are also classified as reptiles. And crocodilians, or alligators and crocodiles and caimans, are also classified as reptiles. But what I want to show you is interesting is that this is kind of a really crummy name. It's almost like reptilia is like the protozoa of birds. It's like a dumping ground for organisms. If it's cold-blooded and has scales and lays eggs, we call it a reptile, right? But it's really a bad grouping name because they're really not that closely related. So check this out. There's some skull features that reveal that the class Reptilia is really a crummy group. It's not going away because everyone from kindergartners know what a reptile is. But the class Reptilia includes organisms that really are not that closely related. So if we look at turtles, we have a turtle skull here. This is a snapping turtle. This one has a letter B on there, so it must have been on a test at the time. This turtle skull is what's called an anapsa skull. So let me go ahead and draw what that looks like.
Looks like something from like a world building.
So
Someone made that.
Go ahead and do it over here. So, anapsida
I like those little fish.
skull. And what this basically is this would be the turtle position. Only turtles have this feature. Turtles appeared in the early Mesozoic at the Permian, Triassic boundary, basically right when the Triassic happened, turtles kind of first appear as marine organisms. But what we have going on here is a skull that looks something like this. So here's kind of the turtle condition. The muscles that articulate the jaw, the muscles that kind of open and close the jaw, are all internal. So they originate from the back of the cranium, the back of the skull, and then they go internal into the skull before they attach to the jaw here, articulate. So there is no temporal opening. So you can see this, my drawing's kind of crummy, but what you can see here is on our turtle skull, from the side and from that one there, you don't see any external, there's no muscles that are on the outside of the skull. It's basically kind of like an armored tank. These things kind of have the bones on the outside. The muscles that articulate the jaw, you can actually put your fingers in there and do this. My finger is sticking out where the muscle would be. They attach here at the back of the skull, and they go internally and connect to the jaw inside. And so it's all covered by this big shield here. And so I guess I'd say the jaw muscles are very fish-like. in a fish, they kind of operate the same way, where the jaw muscles are kind of like underneath the skull there. And so these are, I don't know, I guess you'd call it internal muscles in the skull to articulate the jaw. You can see where the muscles attach on the skull because they're kind of spongy, where the muscles would have attached. But this is what's called the anapsida condition. There's no temporal opening. I'm drawing the muscles as dashed lines because they're actually inside of the skull. So, but lizards, this is a monitor lizard skull, and crocodilians, this is an alligator skull, are what are called the diapsida condition. So I have an alligator skull here that shows this, and there are openings for the jaw muscles to pass through, and there's two of them. So let's go ahead and draw this, the diapsida skull. And this includes reptiles plus crocodilians plus birds. Okay, and how this skull looks is something like this. Here's maybe like a reptile example here. Superficially kind of looks the same. There's our jaw. Here's our eye opening. And what's going on here is there's actually two kind of lobes or openings in the side of the skull where the jaw muscles, and there's two pairs of jaw muscles, come down and attach to the jaw to articulate the jaw. So here we have two temporal openings. Or lobes. It's not super easy to see. You can kind of see it in the crocodile or the alligators here. There is the two temporal openings right there. There's two openings here. And what happens is the jaw muscles originate from the top of the head. They're exposed. They pass through these openings. This is like a cheekbone here called a zygomatic arch. And then the muscles attach here at the jaw. The front muscle would attach there, and the back muscle would attach there. And so they actually have two pairs of muscles on each side of the head, but they pass through two temporal openings to get there. And the same is here for the monitor lizard. There's one opening here and another opening there. So, birds actually have the same feature. So here is the class Aves, which these are diapsids also, and this is a pigeon skull. You can see that there is basically, this is kind of like one of my drawings. There's two lobes here where the jaw muscles attach to the cranium, the skull, and then they pass through these two slots here and attach. There's one attachment point there. There's a second attachment point there. So reptiles, lizards and snakes, crocodilians, alligators and crocodiles and caimans, and birds all have this two temporal opening feature. What's even more interesting is that alligators or crocodiles and birds actually have even more in common than crocodiles have with lizards. So in crocodilians, they have two additional openings. They have a cavity or an opening in front of the eye called the ocular foramen, and they have a mandibular foramen, a jaw opening. These foramens are for nerves to exit or basically come from outside from sensory cells to go into the brain. So don't worry about the name foramen. Basically, there is an opening in the jaw and an opening in front of the eye on the crocodile skull, or the alligator skull here, that the lizard doesn't have. So the lizards and snakes don't have this. They don't have a opening here on there or on the jaw. The crocodilians have this, and birds have it as well. So birds have a opening in the jaw and an opening in front of the eye, opening there. So that would basically say that the crocodiles and the birds actually have more in common with each other in terms of their skull features than either of them have with the reptiles. And none of these have anything in common with the turtles. So what we consider to be reptiles, which includes lizards, snakes, and turtles, is really kind of like a bad combination. So there has been a push in vertebrate biology to rename these things. And that would be, the turtles would be in a group by themselves called the testularians. Let me go ahead and draw a little diagram here. Basically, this is what should be probably the case. Is we would do the turtles, and I'm not going to ask you these names, but this would be called the testularian. And test means shell. We've heard that before. This would be the lizards and snakes, and that would be called the Saurischia. For Saurischians, Saurischian means reptile in Latin. And then this would be the crocodilian and birds. And this group would be called the Archosaurian. Because they also have the most in common with certain kinds of dinosaur skeletons, namely the ornithischians, which are the bird-like dinosaurs. So this would be a more appropriate classification, but to call these guys turtles, lizards, snakes, and crocodiles reptiles, and leave the birds out, we know that goes against cladistic classification. That's kind of an illogical grouping. But anyway, this probably will take a long time to happen if it ever happens at all, because we've been calling lizards, snakes, crocodiles, and turtles reptiles for so long, it's hard to make the adjustment. Okay, so let's go ahead and look at some other features of the aves. So I think for the test, what I'd like you to do, I'd like you to know what a turtle skull, the anapsida skull is. Obviously I have a label on this, so it'll help you know this in that way. So you should know that turtle skull is anapsida. Currently, turtles are classified as reptilia, so we'll just go ahead and keep that. But I think you should just know that it's likely that name is not very valid. We're just putting them there for convenience because that's what we've learned, for a long time. Lizards and snakes are also classified as reptiles. I don't have a lizard or a snake skull. I have a snake skull back there, but it's really hard to see the features. But they basically have the same things in common with crocodiles and birds, which is they have a diapsida skull condition. So for this alligator skull, you should know that this is the diapsida skull, and that's also classified as a reptilia currently. But the birds are also a diapsida skull. This is a heron skull, and this is cool because you can totally see where those two muscles go on this one. The muscles basically where the black pen is one muscle, and where the green pen is the other muscle. So you can see those diapsida conditions, basically the two temporal lobes here are present. And this one I have labeled C. So basically the birds are diapsida like the reptiles are, A, but the turtles are not diapsida, they're anapsida. So they're a totally different kind of thing. So what on the test you probably will need to compare the turtle, the crocodiles or alligator skull, and the bird skull, and be able to recognize anapsida versus diapsida skulls. On Thursday, we'll look at the mammal skull. The mammal has what's called a synapsida skull. You only have one temporal opening. So you have one jaw muscle that goes from your cranium basically down to your jaw. We'll take a look at the synapsida skull on Thursday, so we don't confuse things here. I have some reptiles and snakes. Basically, these are classified as reptiles. They have scales. They lay amniotic eggs. They have some heart features that are very similar to the amphibians. So we'll get to that. But what's interesting is that the crocodilians, the alligators, the crocodiles, the caimans, and the birds actually have a lot more in common. They have a 4-chamber heart. They have semi-warm-blooded. They actually have the ability to do endothermy. They can regulate body temperature internally with hormones. They take care of their young. The birds will basically take care of young in the nest, very similar to how crocodilians will take care of their young in their nest. So there's a lot of features in common between the crocodilians and the birds, which is why that group, Archosauria, should be used. In fact, the scales of reptiles and crocodilians are very closely related to the feature of birds that we're familiar with, which are feathers. So here's a link if you want to check it out. That's an active link on the PowerPoint that's posted. It's a paper that basically shows that scientists have been able to actually modify genes of scales to turn them into feather-like structures. So it's basically a scale modification that turns it into a feather, kind of extends it into a compound feature. If you've ever had birds, you know that feathers start out as a pin feather. And pin feathers are basically very similar to like the scales that you see on the throat of some lizards. That pin feather then erupts into a downy or plumy type structure. So feathers are basically just modified scales. So on the exam, we're not going to do too much with birds here. I mean, they're super interesting and complicated, but we're just going to consider it basically like a feathered crocodile, right? Basically, so it's a, here is our class AVs. It'll probably be that quail that'll be on the test. And so you should know that's class AVs. And that the feathers on there are basically modified scales from the reptile, exaptation scales. Okay, on Thursday we're going to look at mammals, so we'll come back to the mammal region there. I think that gets us some good questions for these, and we'll look at the amniotic egg on Thursday as well. Let's go ahead and look at the dissection of the frog, and that'll get us for today. So we have these frogs. They're preserved in Curacao, so we have some gloves if you need them. So again, wash your hands after you're done, use some gloves. I also appreciate last time everyone was super good. You washed the tabletops off after we're done. Let's do that again for these. For this dissection, I think we're going to need to use these trays. So we have these kind of wax trays because they're a lot smaller than fit the frogs. So we'll use those. They're back here. And again, when you're done, we'll clean them out. Throw any cuttings or scraps that you pulled off the frog into the trash, and we'll put the clean hands into the sink, kind of lean back so they drain. Okay, so for our frog dissection, we're going to have a couple things here. I'll show you the key things we're going to look for. Cut it like you did with the shark. You're going to cut it on the ventral side from the pelvic opening, the cloaca, all the way up to the rib cage. There's actually kind of a sternum here, or a pectoral girdle for sternum. You're going to have to cut through that with some heavy scissors. I would recommend using the heavier scissors for this section versus the little fine scissors because you'll get a little bit more leverage. So okay, let's go ahead and I'll draw our frog here. The ventral side of the frog. And you're cutting from the pelvic opening, the cloaca, all the way to the sternum, basically. Here's our frog here. Okay, so here is the mouth there. All right, so you're going to go ahead and make an incision with your scissors. You don't need a scalpel on this one. Scissors here all the way up to between the arms and as far up as you can get. You'll want to see the heart structures here. All right, let's go ahead first off and look at some internal organ features. And for internal organs, we have, let me get the right pen here. We have a liver, basically, that will be sitting right here. The liver is actually, on this page, it's 3 lobes. It has kind of three primary lobes here. So we have our liver. In this case, the liver should have a gallbladder on it. And I know on our sharks, the gallbladder was kind of messed up. So we didn't have it there, but the gallbladder should be present on these. And that is part of the liver. The gallbladder, again, secretes like a detergent, like salt, onto the food as it passes through the duodenum to break up fats. So gall or bile is basically a fat emulsifying or fat breakdown compound there. So the liver basically separates our 2 cavities. Our peritoneal cavity, which is our gut cavity, abdominal cavity is below the liver, and the pulmonary cavity, which has the lungs and the heart in it, is above the liver. There's no transverse septum like there was in the shark, or diaphragm like we'll see in the rat. Okay, so the gut features, what's really cool about vertebrates is they have all the same basic parts. The esophagus comes from the pharynx behind the heart and lungs, and so you'll see basically the stomach will be here in the same spot that it was in the shark. The stomach is kind of off to the left side, and it kind of has a bit of a, like a kind of an elongated bag. There'd be a pyloric valve here, which is the junction between the stomach and the duodenum. And then the duodenum is this, it's very characteristic. Every vertebrate has the same basic feature. It's like an S curve. From the stomach to the small intestine, the ileum, the duodenum is like this kind of S bend in the gut. Or duodenum if you're American. Okay, so then let's go ahead and go with the rest of it here. The ileum in this case, it's actually very, it would be considered a small intestine, meaning it's small in diameter. The ileum is kind of a convoluted, we have kind of like this long spaghetti-like tube that is the ileum. This. Okay. And this is the absorptive part of the intestine, the part that the food passes through and absorbs nutrients from the food. You'll see a lot of vascular tissue on that for absorption. And then there is a very, abrupt, short colon, which, in the rat we're going to call it the large intestine, but it's basically the colon is the water resorbing part of the intestine. It's very short. It's only about maybe an inch long in the frog. So those are the primary gut features: stomach, pyloric valve, duodenum, ileum, and colon. I think the only new feature the shark didn't have was the colon, right? The shark didn't have a colon, but the shark had a rectal gland, which, the frogs don't have. Okay, so internal organs that you need to know would be associated with the duodenum is the pancreas. The pancreas is a gland that is kind of surrounding the duodenum here. So this is the pancreas. I think it's fascinating that pancreas is so kind of glandular and amorphic because, people have pancreas cancer and people have their pancreas removed. It's like this really amorphous glandular thing. It's kind of amazing or a miracle they can even do work on that. And you can see that it's the same spot. It's really kind of hard to, sometimes it's difficult to identify, but for certain I'll have at least one organism pancreas you have to identify. The other one to note here is the spleen is located just below the stomach and the duodenum. I think on the shark, the spleens are kind of, you know, maybe elongate or maybe a little triangular. This one's going to be more like a raisin or like a cranberry. Thanksgiving was so recent. The spleen looks kind of like little cranberries. That's going to be off to the left side, underneath the stomach, basically, is where the spleen is located. Okay, and then we have kidneys. In this case, the kidneys are no longer embedded in the back wall. They're kind of more pronounced into the body cavity. So the kidneys are here. And they're more developed. As you can see, they're kind of out into the body cavity. They're no longer embedded in the back body wall. And these, because these organisms are more terrestrial, the kidneys are taking on a role of absorbing water or retaining water in the body and concentrating urine for any nitrogenous wastes. Whereas in the shark, the kidneys were for retaining urea into the body. So it's a totally different function. Okay, I think that gets us all of our peritoneal organs. So stomach, pyloric valve, duodenum, ileum, colon, fair game, kidney, spleen, pancreas, liver, gallbladder. That's pretty much what you're going to see in the peritoneal cavity. There might be gonads to wade through. If it's a female, she might be loaded with eggs. And then I'll have to come around and show you. The eggs will be associated with fat bodies, so you can pull those out. And then probably you'll see ovaries and testes. Again, the ovaries and testes are up here underneath the armpits, like they were in the shark. Some of you were surprised that the testes in the sharks were kind of up by their pectoral fins, but the same kind of thing happens. All right, let's move up to the heart structure. In this case, we have a 3-chamber heart. The heart is located here, and the ventricle is the same, but the heart is what could be described as a 3-chamber design. We have a ventricle. I'll put a V there. I'll draw a heart here more elaborate, but the ventricle is there, and then rather than having a single atrium with a sinus venosus, we have 2 separate atria. And so what that looks like is there is a left atrium, which is probably homologous to the atrium that occurred. Let me go ahead and do an LA for left atrium. And this is frog left. I know it's your right. It's on the frog left side. And then we have a right atrium, which probably what happened was in the shark condition, we had the atrium on top and the sinus venosus below. That probably twisted clockwise, and the atrium from the shark became the left atrium, delivering blood back to the ventricle, and the right atrium basically probably derived from the sinus venosus, which receives blood from the body. It kind of makes sense, you know, plumbing-wise. But it's a 3-chamber heart with a left atrium, a right atrium, and a single ventricle. That's why it's called a 3-chamber design. Let me go ahead and label that left atrium. I want you to see these. I possibly will have a pin in those on a test and ask you to differentiate left versus right atrium. Okay, and then B here for ventricle. I'll put an arrow to it because it'll cloud it up. Okay, so the big difference here, the reason for the 2 atria is the right atrium receives blood from the body, from the jugular veins and from the cardinal veins, the main veins of the body. Delivers blood to the heart, The heart then pumps the blood back to the body, and one circuit pumps it back to the lungs. And so the big difference here is these organisms have lungs. And the lungs are located just behind the heart here. And these are a respiratory adaptation, probably homologous to the swim bladder that occurs in fishes. But I'm going to argue next week that lungs probably evolved first, and then swim bladders evolved later. But anyway, the lungs here basically receive blood from the third circuit of the heart, and then deliver the blood back to the left atrium with oxygenation, or fully oxygenated. So the left atrium receives blood from the lungs and then dumps that into the ventricle commonly. So it's kind of interesting, the plumbing wise, is there's three afferent branchioles that branch off. The first ones go to the head, the second ones go to the body, and the third one goes to the lungs, and then they all mix together in the ventricle. It doesn't really make any sense why you would only send 1/3 of your blood to your lungs until you realize that frogs and amphibians have a very, very thin skin that has vascular tissue. They can actually do a lot of their respiration through their skin. They don't need lungs per se exclusively to do respiration. They can do maybe as much as half of their respiration through what's called cuticular respiration through their skin. That's one reason they say that when you're handling frogs and salamanders, having lotion or soap on your skin can really be damaging to them, because their skin is so thin and has lots of vascular tissue. They're doing a big bulk of their respiration is actually through their skin. Less than half of the respiration is probably through lungs. This can change. For example, toads do most of their lung respiration through lungs, but there are some salamanders called lungless salamanders that don't even have lungs, and they do all their respiration through the skin. So what we're seeing here is kind of a transitional time where the three-chamber heart design is kind of adapting to doing lung respiration. And then we're going to see on Thursday how the three-chamber heart design is modified to divide the ventricle up so that all the blood goes to the lungs. But this is kind of our transitional time. Three-chamber hearts occur in amphibians and in the lizards and snakes and turtles. Turtles, lizards, and snakes, and amphibians basically all have a three-chamber design. The crocodilians and birds have a four-chamber design, as well as mammals have a four-chamber design, but they're built backwards from each other, so they're not homologous. They probably happen separately. Okay, I think this would be enough for our dissection. So be sure you dissect up enough to see the heart, because I would like you to differentiate left atrium versus right atrium. Ventricle would be the same as the shark, and then the lungs will be an important thing to see behind the ventricle. So at least on the frog, if not the rat, the lungs could be a possible question on the rat. Sound fair enough? Not that bad. Also, even though I'm not going to have them a test, be sure you check out those temporal tympanum, the eardrums, because they're external on the frog. If you cut open the tympanum, you can actually find that little stapes in there. It's super tiny, but you'll see a ligament attached to it. It's kind of like a little string on it. So if you can find that stape, basically you found the inner ear bones of the ossicles on the frog. Okay, so how we have the frogs going on here is, let me get some gloves on, and I'll show you where they are. You can get your own trays and your own tools. And the frogs are in bags. When you're all done with the frogs, we're going to go ahead and return them to this big bag here, because I'm going to use your frogs on the test. So if you have a really good frog dissection, you may see your frog on the exam. So here's our frogs here. They're individually wrapped. Christmas comes early for everybody. Here you go. So anyway, I'll go ahead and I'll put the frogs right here.
You know, I was actually grabbing a big one out of this bag.
Maybe not raw, but
Big one.
we can microwave it in Mesa.
Go ahead and pull those out. And once you